My group does two what sounds like very different things, but they'll come together in this talk and in my research. One is the chemistry part, or the measurement science part. We develop new tools to study the chemistry happening in very small samples. And the chemistry are all the molecules in something the size of a single drop, or even a millionth of a drop. And so this is measurement science. And so it's a cross between chemistry and bioengineering. And it has all sorts of fancy terms up here, depending on what the molecules are. I have a quote here that sort of guides me, and that is, really, since telescopes and microscopes and things, as a new tool comes up that gives you a new view of looking at the physical world, we tend to learn more about it. And there's probably no field that's been more true in than studying the brain. That is, advances such as magnetic resonance imaging or microscopy or others, as they come into play, we learn more about how the brain works. And so you'll see that theme today. So this is one of the things I want to say, and this is just simply a listing of some of the affiliations I have because of my work being in between those two fields. The other thing I do, which I'm not going to talk too much today, is I do run something that's called the very fancy, lots of big words, Neuroproteomics and Neurometabolomics Center on Cell-to-Cell Signaling. It's funded by the National Institutes of Drug Abuse, because if you study the chemistry that's happening in the brain and health, if you study the chemistry of molecules like serotonin or dopamine or peptides that hit opioid receptors, those can also be abused. And those are the exact same receptors and molecular systems that drugs of abuse work. And so my group has helped to run with collaborators a center that for the last 16 years. And there's a website there that lists a lot. So I'm not going to talk as much about that, but if you listen to today's talk, or you have any questions about that, I'm certainly willing to talk about that. The other thing I want to do is, it's very easy for me to stand up here or sit down, I guess, in my room, my house, and talk about this research. It sounds like it's work that I do. It's a large team. I'm going to be showing you some results going back about a decade to some very current work. And this is showing you some pictures going back, these are a couple years ago, to the one in the lower left was in January before we went into the COVID lockdown. When I see these pictures now, it looks a little strange because everybody's close together. That's not the way the lab looks. But these are the people who are doing the work, and then lots of collaborators. And so it's important to give them credit, and I'll try to name people as I go. But again, it's a team effort. So, from a fundamental question in health, what is memory? And, you could even ask something more complicated, or, maybe even simpler to ask, and that is, what is consciousness? If you look, Aristotle made some attempts to understand memory, and he actually said that, the way memory works is wax tablet, and your experiences write little marks on it, and somehow you can read those out. And what's amazing is, even though it's been studied for a couple thousand years, if you want to count that, we're still trying to address that. Where is memory? Where does it go in the brain? Is it the connections between the neurons? Is it the chemicals between them? And how does that actually work? And so what's really kind of fun is that in the areas of chemistry, in microscopy and others, we're actually getting to the point where we can actually address this in a complicated organism, in a way that we never could before. Today's talk will mostly fit on the chemistry. This slide just shows, 4 different views of a brain. This happens to be showing a human brain at a scale where you can see the cells and the connections. The artist that drew this made glowing white, glows as the signal moves down, indicating something that's happening. A lot of people tend to think of this as electrical, but it's also electrical transmission until it gets to the next slide, next view, which is now synaptic chemical connections. And in between two different cells, you have the flow of information that actually is converted from this electrical chemical gradient to chemistry. It turns out, if you want to think about it from a different point of view, and that is, what happens if something is not working in a brain? How do we fix it? We're unlikely in an adult going to be able to rewire it. But what we can do is change the chemistry. either with pharmaceutical intervention, diet, and other things. And so again, we want to understand the chemistry that's happening in these small domains and in the brain and how those relate to behaviors. So again, simplified pictures of what a neuron in the brain looks like. And again, you have 100 billion of these, is you have a cell body, it sends the message down to a release site, and then it releases a host of chemicals. And this is a picture that a lot of people think about, but a single neuron could actually have 1,000 inputs from other cells to it and many outputs. And whether or not it fires depends not just on what's happening at the synapse, but everywhere along this. And so this chemistry is complex. It's not digital like a computer, but it's actually a very complex chemical soup that these neurons are bathed in. And my group is trying to understand what that chemical soup is and how it changes. And the other aspect of this is, hopefully what I'll show you today, is that we don't even know in some cases. We're still discovering new molecules as we look at this. So here's sort of that's the neuroscience background, and then I'll get to a little bit of chemistry and go back and forth. This picture is just showing you, some of the neurons. The other thing to point out is when you see pictures like this, many people tend to think of this as, sort of an accurate picture, but there's no empty space in your brain. These neurons aren't floating in a network in space. It's filled with other cells. And so I like to sort of joke and say the font of all human knowledge, Wikipedia, claims that a microliter, that's a very small, a millionth of a liter of mammalian brain, has 100,000 neurons and about 400,000 other cells. And so if you think about this, this empty space is filled with all the helper cells, the astrocytes, the oligodendrocytes, even red blood cells. At any one time, there's more red blood cells in your brain than actually neurons. And most people don't tend to think of that. So if you were to take a rat brain or something else and count the cells, you would say, wait a second, it's mostly not neurons. And that's because of all these other cells. And so it's not empty space. And then at the end of this talk, you'll see how we try to address that by going to the single cell level. So here's a picture of a cell. And what I'm trying to show on this slide is the comment that we can actually do single cell analysis in some cases for some of the things like RNAs and DNA. We know a lot of the proteins. We actually don't know all the small molecules. One interesting, and some people don't like it when I say this, but one interesting way to think of the brain is it's a bar of butter. And what I mean by that, just like butter, is it's mostly water and fat with a few percent of protein and even less of RNA and DNA. So again, there are more lipid molecules in your brain by far than all the rest of the other types of molecules outside of water. And they're actually dynamic and changing, and lipid maps, an international consortium, now says there's 20,000 different chemical entities that are distinct lipids in your brain. And we don't even know what all of those are and how they work. And so we're working on that. I'm not going to go into much detail, but there is some evidence that as those lipids change in a bad way, you can actually get some diseases, but there's no easy way to study that. And so it's been sort of an overlooked part of the brain. But I also can say that if you tell somebody their brain is like a bar of butter, they tend to think you're insulting them. So that's maybe not the right way to say that. So let's start with what most people think of when you say chemistry and brain. They think of neurotransmitters. Most people who don't study the brain still know about serotonin because of the fact that serotonin is, known to be or thought to be misregulated in different forms of, for example, depression, dopamine, and anxiety. Dopamine is also known as the reward molecule. These are neurotransmitters. There's GABA and glutamate, and then a whole host of peptides. And so these are just a slide showing these. We tend to think of these in a very human-centric, and so I'm going to sort of be a little bit different and show you some work we're doing on other animals. It turns out, this is showing the metazoan, the animal kingdom. Every one of these animals, basically, or most of them, use the same neurotransmitters. They all use serotonin and dopamine and acetylcholine and all the others. And so the neurons may be just as complex as ours. What's different, of course, is we have more of them. And so what I would like to show you a little bit is some of our work to characterize the chemistry in the brain in a whole host of different types of brains, and each one will have advantages. And this is just showing you some of them, and I'm just going to talk about a few of them. For example, I'm going to show you a sea slug that can eat another slug whole, a Pleurobranchaea, but if you feed it a Spanish shawl, this is purple and orange. This is underwater. It's a sea slug. You can't see the water too easily. It will get stung in the mouth, and it will remember what not to eat. It won't try to eat a Spanish shawl again. Where did that memory go if it only has a couple thousand neurons related to feeding? And then we work with a large number, and this is sort of a joke. That's not quite the way a cow would smile. But anyway, so I'm going to show you this and tie them together among different brains. The other thing is that oftentimes we work across scales. This is a honeybee brain, so work with Gene Robinson, the director of the IGB, or was until he's now the Liberal Arts and Sciences interim dean, to a brain slice, to a single network of neurons. And we work across scales to know where the chemistry is that occurs in this network of neurons in the center picture. For example, where is serotonin, or where is acetylcholine? When is it released? This network of neurons actually controls, or would have if it was in the creature, the reproductive network of in this particular case, the animal that we were working with, Neuroproteomics californica. So these are the types of systems we work with, ranging from single cells, simple networks that are still complicated, to entire brains. So let me start with one of the simpler animals I'm going to work with. This is Neuroproteomics californica. It's a sea slug. It won the Nobel Prize. It didn't. Eric Kandel got the Nobel Prize 2 decades ago for his work in learning and memory in a sea slug. So when you say learning and memory in a sea slug, you're not talking about the learning that you're doing in this talk, but you're talking about much simpler associative learning. And let me show you an example. One reason we do this is the cells are big. Most are not, but some are. This is just a giant neuron. So if you're going to study what's happening in one cell versus another, they're much bigger than most. And so they're typically on the order of 50 to 500 microns, much bigger than ours. And this makes it easier to work with. And so there's 10,000 neurons. It has 1 million synaptic connections. Its genome is the same complexity as ours. It's almost, it's 3 gigabases about. And so its chemistry is complicated. Its brain is much simpler. This is one of the reasons we started working with this. And this is an example of why Eric Kandel got the Nobel Prize. And I just show you this because this is the example maybe of the simplest memory forming network. This is 2 cells shown representative, a sensory neuron and a serotonergic neuron, motor neuron. And so here's one I'm showing here. It happens to have a name, L7. It's on the left side of the brain, the 7th one characterized, and a sensory neuron, all these different connections. And so this is just really 2 cells. This is the nerve terminal. And you can ask a question. You can actually stimulate electrically, and this will remember until the dish dies. And you can reset it with serotonin. And so what changes even in a simple memory forming network? And so this is probably the example of getting memory to its simplest. And then, of course, more complex systems can be studied. So let me show you more of a real life example. This is Pleurobranchaea californica. It's a predator. And I like this one when you're sitting listening to me at 5:30 before dinner, because this is something about feeding thresholds, something you can measure. So Pleurobranchaea is a predator. It eats almost anything. If you want to remember some funny fact that has nothing to do with its brain, if it's attacked, it secretes 1 molar sulfuric acid on its skin. And so that's a little bit, if a fish tries to eat it, it has a defense, and that is it'll burn the fish's mouth. And so that's a chemical defense that's kind of interesting. But here it is about to eat something. That would be scary if you were a small animal. What we can do is go into the brain and decide, the part about this that's interesting is the most dangerous thing that a Pleurobranchaea does is it eats. The most important thing it does is it eats. The reason it's dangerous is rather than being in a crack, it has to come out and prey, and that means it's vulnerable to predation. And so it's doing a cost-benefit analysis to figure this out. Rayner studies that. But the other thing that's interesting is that if you feed this, it will, and then you feed it again, and it'll stop eating. But if you give it something, for example, butane, as a stimuli, it will still eat. Now, that's a good example because it's the same reason that when Thanksgiving comes out for some of you, at least in my house, and you eat too much, you stop eating. But then when the pumpkin pie comes out, well, you still eat. You don't need to eat, but you still do. And what determines whether you eat or not are these thresholds. And it's actually an interplay with serotonin in every animal. It's the serotonin in the enteric nervous system that deals with that. And even in a Pleurobranchaea, that's true. And so we wanted to understand that. And so what we do is we came up with a way, and this is showing now a Pleurobranchaea to an aplasia. We can take a single cell out, we can isolate it, we get it in a little microdroplet. And I'm just going to show you one slide of one of the things we do. We do something called capillary electrophoresis, where we put 20,000 volts across a capillary. The cell blows up and or ruptures, and we can measure the chemicals that are in the cell based on their electrical migration in this capillary tube. And when they fall out, we can detect them with either laser-induced fluorescence or mass spectrometry or another approach. And so what we get is an electropherogram showing things like serotonin or dopamine or others. And when we did this, and this was over a decade ago, with the one cell that we thought was a command cell for feeding in Pleurobranchaea, we saw that hungry animals, in this particular case, had higher levels of serotonin compared to full animals. This was the first case where a neurotransmitter was, in a single cell was linked to an overall behavior. And, you can see there's a lot of variation because these are wild-caught animals off the coast of California. These are not, laboratory-raised animals. They're shipped from California to Illinois. They're probably not very happy by that. And then we're able to do these feeding experiments. But there is a robust change in the chemistry in one cell that then innervates the entire feeding network. And so we also use serotonin in our enteric nervous system and to control, and so do a lot of mammals. But this was, a start to trying to understand that. One thing to say is serotonin chemistry is complicated. I will say what I am showing you here and on the next slide is a special system my lab built using this laser-induced fluorescence, using a UV laser that enables us to detect just the indole ring of serotonin and other molecules better than maybe any other system. And so we can go down to the attomole or even in some cases the zeptomole levels of serotonin and any other indole. And so this is showing you serotonin, this is proteins with it, and there's hydroxyindoleacetic acid. If you wanted to see how complicated this can be, this is serotonin catabolism in a mammalian brain. And I'm just showing you this because this is us discovering, here's serotonin and tryptophan and tyrosine and hydroxyindoleacetic acid. And this peak right here is a new serotonin catabolite. It's the indole ring right there, forming a product with cysteine. So it's 5-hydroxyindole-thiazolidine carboxylic acid. And this is found only in certain conditions in mammals. And I'm just putting this up because chemistry of something as simple as serotonin can get complicated. It's sort of a 2-slide little blurb saying, I'm not going to go into this, but this is one of the, aspects of the, the work with drug abuse. And this is just showing you at a different change of scale that if you induce serotonin syndrome, you actually can make and see lots and lots of changes in serotonin catabolism. Not necessarily important for feeding, but it does show you how even something as simple as a serotonin pathway, when it's overloaded because you're taking a drug like ecstasy or also on an antidepressant in this particular case at the same time, this is in a mouse. So we've induced both. You can see serotonin sulfate, this new compound. So one pathway explodes in complexity in different parts of the brain. But let's get back to something simpler. Serotonin is very well studied. Every catabolic pathway in serotonin outside of the one I just showed you is the site of a multi-billion-dollar drug, and there's thousands of papers. So I'm going to show you something that's sort of different, and that is going to go to the amino acid transmitters, but I'm going to do this in a very weird way. So this picture is showing Alice in Wonderland through the looking glass because everything's reversed on the other side of the mirror. And what I want to show you is some interesting work that we're doing on d-amino acids. And so L-aspartate is an amino acid that most people think is endogenous. It's in the food you're eating. It's used to make proteins. D-amino acids are in microbes, but most people tend not to think of them in higher animals and certainly not in our brain because we wanted that to be this, the ultimate and higher organism. And so they're hard to measure because they're identical except mirror images. And so again, outside of glycine, all amino acids have two forms, the L and D form. And we tend to say, if you look in biochemistry books, life uses L amino acids, at least higher animals. Protein synthesis only uses L amino acids. Sugars are in the D form. It's how they rotate light. But there is evidence that there are some d-amino acids in the brain, and they're there for good reasons or bad. This is not what I'm going to talk about, but it's one that everybody seems to be learning about, and that is there's a d-amino acid that's been known for about 15 years. We've been studying it. We're working with a lot of people studying it, and that's D-Serine. It's the optical or mirror image of L-serine. And I picked this slide because it shows something, and that is D-Serine. It correlates with schizophrenia. The first groups, they're friends of ours, friends of mine, for example, Jean-Pierre Mothé and others that started studying about this, were looking at how D-Serine hit the memory receptor, NMDA receptor in the brain, not thinking, they're studying something fundamental. And it turns out later on, it really was shown, and it's still being shown, that D-Serine, its mode of making or its destruction through d-amino acid oxidase correlates with schizophrenia and some other mental diseases in humans. I put this up because correlation is not causation, and there's lots and lots of papers that say it causes, for example, schizophrenia. It doesn't. There's no evidence that it causes. It's misregulated in schizophrenia. And the real purpose of D-Serine is not to cause a mental disease, but to actually, it's involved in memory through the NMDA receptors. D-Serine is a GRAS product, generally recognized as safe. It's something that's made in fermented food, like whether you had kimchi or vinegar or sake or something that's, some alcohol, there'll be a lot of D-Serine in it. And because of that, it's not regulated. You could go to the local nutrition stores, wherever your favorite one is, and buy D-Serine. And I only say that because some colleagues of mine at the last meeting I went said the biggest problem is that the way the body gets rid of D-Serine, the neurons get rid of D-Serine, is it oxidizes it and makes peroxide. And if you take too much of it, there's some evidence that it can kill the cells that are trying to do that. So this is not necessarily, even though it's sold, a safe product at thousands of times the natural level. So if you see this as a supplement, take low amounts and not the most you can, because there's some evidence this would not be good for the brain. But I'm going to talk about D-aspartate. If you were to look it up, most people would not say it's a neurotransmitter. It's found in all animals, from humans to frogs to others, that's been tested. But it may be a neurotransmitter. So I'm going to show you in 5 slides, a 10-year attempt to show that D-aspartate is a neurotransmitter. Again, it's the optical mirror image of the L-aspartate. So going back to that weird approach of mine, capillary electrophoresis, this is just showing you time and peaks, and there's a separate peak for D-aspartate and L-aspartate. We can separate the two because they interact differently with the walls. And in aplesia, that sea slug I was showing you, we found some neurons with lots of D-aspartate and a little less of L-aspartate. The problem is, even though D-aspartate is found in humans, it's found in rats, it's found in sea slugs, there's no enzyme that makes it. There's no gene you can look at. You can't use genetics to do it because people don't know yet what the gene is that makes it. So if you don't have that type of handle, you can't use RNA or DNA. You can't make an antibody to localize it because, the enzyme, because we don't know what it is. So we picked a simple animal and we said, can we figure that out? And there's really high levels in parts of the brain and lower levels in this sea slug and other parts. You can even go to a single cell level. Our approaches work at this small size. This is the size of some of the bigger cells in your brain, and there's lots of D-aspartate, and it's transmitted, transported down a process. We used a retro experiment. If we feed these cells radioactive L-aspartate, they make D-aspartate on the left. If you feed them radioactive D-aspartate, they almost make no L-aspartate. If you heat the cells up and kill them, they don't do this. And so it seems to be an enzymatic pathway, even though the enzyme isn't known. If you take, in this particular case, 5 animals and look at the L-aspartate released in a potassium-dependent manner, some animals only released L-aspartate. This animal mostly released D-aspartate in the cerebral ganglia. And so everything suggests that these cells can make D-aspartate, transport it to a release site. They can release it and After a little bit of work, we even finally discovered the first mammalian, or I'm sorry, the first animal in any animal, D-aspartate racemase. And this is an L-aspartate, and this is just showing you the work. And this was, and so D-aspartate, this in this very small snippet, is a molecule that may be a neurotransmitter or a hormone. And I'm not going to go into too much more detail on this, but D-aspartate is localized in different animals. I showed you some data from Aplysia. It's synthesized in those cells. It has a potassium-dependent and sodium-dependent uptake and activity-dependent release. It causes electrical activity. And so all evidence on some cells says that D-aspartate is a neurotransmitter and a hormone. And I like this example because it's basically saying that neurotransmitter discovery hasn't ended. Most people tend to think of that was in the 70s and 80s, and now we're at the point trying to figure out what they do. When I show this story, sometimes people say, but what does it matter? Does this impact a disease? Well, if you don't know how it's made or how it's destroyed, you can't even ask that question. So it could be, that there is a disease where this is misregulated or not. We're still looking for that. And most interestingly, and I'm not going to go into this because this talk is in the brain, there's actually high levels of D-aspartate made in the islets of Langerhans, which are related to insulin and glucose metabolism. They're actually localized in the same cells that make insulin. And so it may have something to do with diabetes and the hormone role too. And we're still trying to understand that. But again, this is its use, function, and what it works and does in mammals is really not well known. So when I say D-aspartate is found in a slug, I'm telling you it's found in humans. It's interesting because you can even go more primitive. Most people would go more modern. And this is a ctenophore or a comb jelly. This is one of the animals or the animal that has the simplest nervous system. It actually only has a few behaviors. It has a nerve net. And so this is some work with Leonid Burrows that he published in Nature 6 years ago. And we were asked to figure out what the transmitters were. And it actually has a very simple network of neurons. And when we chemically analyzed these, we actually found that there was L-glutamate and L-aspartate, maybe for protein synthesis, but there was noticeable amounts of D-aspartate and D-glutamate, as well as other compounds. And if you take some of these ctenophore neurons and you record from them in spiking, this is showing you what happens when you add GABA or histamine or acetylcholine. I don't show it, but serotonin or dopamine. The only chemicals that caused spiking in activity were L-glutamate and aspartate and D-glutamate and aspartate. And again, these compounds are present. So again, suggesting that when you go really primitive, these d-amino acids may be some of the original neurotransmitters and neuromodulators. Again, unfortunately, the genomes of these animals, just like humans, are known, but the enzymes, that make this are not known, so they may be misassigned. D-aspartate in Aplysia was actually assigned as a serine dehydratase, even though it seems to be its main function seems to be related to D-aspartate. So this is just kind of, if you were to be thinking about what the transmitters are used in your brain, there are some that aren't very well known. And it's not just D-aspartate or D-Serine. We're working on D-Alanine. It's found in the brain and the pineal gland and a few neurons. I don't know what they do, but this is just sort of going on one step further. There's D-aspartate and D-Alanine in the islets of a rat. And we actually are now getting some human transplant quality islets from an islet transplant center. And there's both D-Alanine and D-aspartate in human islets. And so again, this is some data shown here, but we actually have some new data on humans. And again, whether a rat or a human, the presence of D-aspartate and D-Alanine shows, they're made and released, shows that there is some functional consequence. It turns out both of these tend to change the excitability of the beta cells that release insulin. So they're modulating just like glucagon. And we're trying to understand that in greater detail. So that was sort of a snip of two different systems, one that most people know of, serotonin, and one that many people don't know of, and that's the D and L-aspartate. I thought I would end with the most complicated one. This is just a transition slide. if you're wondering, some nice quotes. And that are the neuropeptides. Neuropeptides are really a complex set of molecules that really are the ones that modulate and regulate a lot of behaviors. And so on a plot like this, it shows, neurotransmitters, it's just one line, but there's actually hundreds of different neuropeptides in your brain that are helping modulate your systems to change your behavior. So this tells you why it's complicated. And that is, this is a complicated slide. You may have a gene to make, for example, neuropeptide Y, change to RNA to proteins, and that would be a prohormone that will eventually be a hormone for neuropeptide Y, and eventually it's packaged and made into neuropeptide Y. The problem is there's so many steps along this slide that there's too many sources of variability. So as you discover a new prohormone, it's very hard to know what the products are. This is a quote that, it's claimed that 4 of the 7 deadly sins are mediated by neuropeptides. And so they do, they are important for behavior. If you were to go to Google and type in neuropeptide or hormone or prohormone, in this particular case that I'm talking about, to make the neuropeptides and the hormones, you get a very different answer. So when you type in prohormone in Wikipedia, you get all these things. This is not what we're talking about. Prohormones here are, for bulking up, but I'm talking about the prohormones that make neuropeptides and hormones. Neuropeptide discovery was hard. In the past, it took a lot of samples. For example, Guliman got the Nobel Prize for looking at one of them, and he claims that it took 4 million sheep brains to figure out enough to sequence a peptide. And he talked about that in his Nobel Prize winning talk when he talked about that. But here's another example. Victor Mutt discovered a lot of neuropeptides, and here's one of his samples. So that's, literally, he was looking at the neuropeptides and he said brains were too hard, so he was looking at them in the gut. And this is pig intestines, full. Horrible sample, I'm sure, to get tons of it. What we do now is we use mass spectrometry, another tool. Sometimes they're commercial and sometimes they're laboratory built. This is at the Beckman Institute. This one is very similar to one that was at the ITB for a long time that's been since upgraded to an Orbitrap. And so we use these. And the reason I show you this is because now what you can do, as opposed to tons of materials, is you can take a single cell. This is my grad students trying to make a picture of this, but you can take a single cell, hit, if you can isolate it, hit it with a laser and you blow it up. Now, what they really do is you put an explosive next to the cell. So when the phaser or the laser hits it, it blows up, and these droplets contain the chemicals, they're ionized, and then you get a spectra. So here's sort of the cell with the explosion, you hit it with a laser, and then you get a mass spectra, and every one of these little peaks represent a peptide that was where the laser hit. And we and a few other groups have really been working on this for the last decade or so, and it works well. And then what happens is this plume of particles goes into a mass spectrometer, like a time of flight mass spectrometer, and then you can measure this mass spectra. So we've gone from requiring tons of materials to a single cell. And because of that, it's a lot easier to do these experiments. And let me just give you a fun example, because I really sort of like picking examples that are, for this, that are a little bit off the wall. You can actually then shine your laser right here and here and here, and you can make a 20 by 20 array. So it's a lousy quality image compared to what you get with a cell phone. But you can then image the spine and see chemicals that are in it. And you can then get a map of what chemicals are present and where they're at. So let me give you a fun study. This is called mass spec imaging using planarian. These are roundworms, and what's kind of interesting is you can cut off their head and they regrow. So these are one of the more complicated animals that actually can do that. And so there's very few times you can say this is after the head has been amputated and 14 days later it's grown a new head. This is some work with Phil Newmark. The question is, how in the world do all the thousands of neurons in the head regrow where they need to be? And this is not very well known. And so we started working with Phil on this, and I'll show you one example. This is with his transcriptomics and our bioinformatics. We discovered 51 prohormones making a couple hundred neuropeptides. And this is some work that was published a decade ago using classical transcriptomics. And this is images saying, for example, we didn't know what to call these, but neuropeptide 4-like. Is located here. This one is in specific cells that are up here. Some are found in the brain or in the eye or in the equivalent to the stomach, other areas, but nobody knew what these did. And so what we did is we did mass spec imaging, and basically you can take a planarian, cut it apart, and you get lots of small ones. Now that's not what we really did. What we did is we took these and cut them across once, and then we looked at the regrowing brain, intact animal, 3, 6, and 12 days. And you can sort of see this where we can do mass spec imagings of particular mass to charges. And you can see this one, for example, is localized to the brain. It's a low quality image, but it's chemical, very information rich. And what you see is, for example, after 12 days, you could re-see the head reform, and you can see the cephalic ganglia, the main head, and you can even see the eyes reforming. And so this allows us to look at the chemicals that are changing during the regeneration of a brain. And this slide is trying to take what actually is tens of terabytes of data into a very simple slide. So we used a data reduction analysis and said, okay, we don't know anything that's happening in this brain. We don't know what the compounds are, but we have thousands and thousands of mass spectra. And we looked at it. This is intact, 3, 6, 12 days after we cut off the head. 12 days, it already has a new head. And we did this dimension reduction and tried to see if we could figure out, in this case, principal component analysis. And basically, all the animals intact are very tightly clustered in this reduced dimensional space. They have the same chemistry. Right after you cut off their heads, you get a lot of variation in the X and Y. So there's a lot of chemical heterogeneity. 6 days later, it's starting to come narrower in one of the dimensions. In 12 days, it's back almost clustered. And so what this is saying a little bit is that every one of these animals that they're doing this have differences in their chemistry, but they start and end fixed with the same chemistry. And if you want to know what that means, you can look at it this way. Each one of these columns is a different chemical. They have a weird name because we haven't named them. And in this one, it's high before. During the regeneration, it's lower, and then it's back up. And these are chemicals, these are peptides that are upregulated during regeneration. And so hundreds of compounds are up and down regulated. And then the question is, what are these compounds? And that's when we went back and we figured out the names and what they did. And so now we have starting to have a feeling of what chemicals control this process. And this is still ongoing research now to figure out how these work and how they interact. And again, this is my group. Paper is listed as Tashuang Ong, but it also is Phil Newmark, who was here and is now at the Madison, Wisconsin, University of Wisconsin. Just as another one slide, we also worked with him, and he basically discovered planarian are like schistosomes, a very close, another similar worm. And he asked us to do some work on why a rotifer could secrete a compound that can paralyze schistosomes and stop their life cycle. And we did not have any idea what this compound is, and why I'm putting it up here is we discovered the schistosome paralysis factor that was doing it is a serotonin-like molecule. So even if you're going to have a paralysis factor that prevents a schistosome from infecting the liver of a mammal, whether it's a pig or a human, in this particular case, not surprising to me, is this a molecule that hits one of the neurotransmitter families. And we discovered this molecule, and we published this just last year. And even at 0, 25, 250 nanomolar, if you take a rat and put it in a tank that has these schistosomes, the right life cycle, they're paralyzed and they can't infect. And so when you look at the liver, there's no infection. And this is just showing you how long they stay paralyzed at different concentrations. But the point is, even when you're looking at something as strange as parasitic invading another species, there are ways to block that. Again, it comes back to things like the same neurotransmitters, because these systems are so important. And I only have a few minutes, and I thought I would end with one last sort of set of slides that sort of explain where we're at now. And that is, we've taken everything I've shown you, and I kind of hinted at this, and we can now do these measurements even at the level of a single cell. Now, single cell measurements aren't new. This slide goes all the way back to Leon Hoek and Hook, who discovered and came up with the first microscope. But most of this period of 400 years, 300 something years, has been imaging, microscopy. And what my group does is really what a few others have done in the last 10 to 15, 20 years is how you can do this, make single cell measurements work at the chemical level. So again, going back to this, I showed you this approach and how it can work at the single cell. This is how you can make this a very powerful approach. This is showing you a group of neurons and one cell is missing. That missing cell, we did mass spectrometry on it. We discovered this peptide here happens to be phenylalanine, methionine, arginine, phenylalanine in there. This cell, when it is electrically active, caused this animal to swim. It's a sea slug. And so we can guess in some cases when we discover a peptide what it does because we know what cell it's in. You can do this in mammals. This is cells dispersed on the glass slide. Every one of these dots is a single cell. We imaged it now, and we can get really high chemical specificity. And every one of these cells, we get a mass spectra, and we can actually see thousands in this particular case of the lipids. And we're looking at lipid heterogeneity at the single cell level. Never been done before. So this was published only a few months ago. And we're starting to look now how lipid heterogeneity changes in development and changes during learning and memory. Just to show you, getting back to hormones and peptides, this is not the brain, but I said, this is now one example of a human, one of the few I'll show you in this talk. This is some human islets, transplant quality islets. And so basically these are collected to transplant into people with type 1 diabetes that have lost their islets. And if they have a few islets left, they'll ship them to us. And so these are, viable living human islets. And these are from individual islets that are supposed to be the same, heterogeneous. And each dot is what we see in a single cell. And in islet number 1, we didn't see much of the peptide somatostatin done with the single cell approach. But we see a lot of individual cells that have, for example, insulin. Islet 4, even more. So we're, so islet 1 had glucagon, but islet 4 has a lot of insulin. So we're seeing for the first time this heterogeneity of individual cells within a microorgan. And we can also do this from the pituitary, pineal gland, and other types of endocrine structures within the brain, as well as endocrine systems like islets. And so again, these are types of measurements that just hadn't been possible in the past. And my last data slide, this is just showing you, because again, you're probably thinking of food and how to escape this talk. This is enteric nervous system has a lot of different hormones that are released when you eat that affect both islets as well as the brain. And here's just shown one cell, and it senses food. It releases, in the gut, it releases hormones into the bloodstream as well as senses them. And there are 6 known hormones. And so the prevailing wisdom was that some enteroendocrine cells release hormone 1 versus hormone 2 versus hormone 3. Of course, that's not what they're called. They're called Gelrin or GLP-1 or PYY. And when we did single cell measurements of this, we discovered almost a combinatorial. So we find some enteroendocrine cells that have hormones 1, 2, 3, and 5. Others have 1, 5, and 6. Others have 2, 3, and so rather than having 6 types of cells, we see that the complexity is that there's literally in amounts and complexity, hundreds of different enteroendocrine cells with different suites of these known prohormones, as well as some peaks that we haven't been able to assign yet. So this is some ongoing work. This older picture is indicating this, so I probably should have moved it over here, but this is some mass spectra where we're just trying to understand this heterogeneity in what was supposedly a much simpler population of cells. And I give this as an example because this takes a very simple part of the sensing part of the system and says, wow, even that is much more complicated than we thought. And again, this ability to probe what's happening in and around individual cells is fairly new. These are just some articles written about it in the last few years. But I sort of wanted to end with some take-home messages. Chemical heterogeneity is high, even in a cell type that shouldn't, that isn't expected to have that, like enteroendocrine cells, beta cells, or if you go to particular types of neurons. It's well conserved across the animal kingdom. Most people are surprised by that. When you think of serotonin and depression, they don't expect serotonin to be in a sea slug. They don't, if you discovered d-aspartate in a human, you're not going to think that it's in a ctenophore, but it's surprisingly well conserved. And even now, when you have a new tool, we're finding unusual and unexpected and novel transmitters and peptides, in the year 2020, and I think this will continue. And that's it. And I thank you very much for your attention.
Awesome. Thank you very much, Dr. Sweedler. Yeah, if anyone has any questions, feel free to either throw them in the chat, or if you want to unmute yourself and ask, go ahead.
Yeah, I like that. Okay, there's a really, does everything apply to octopus's decentralized brains? Well, it's octopus use the same transmitters. I've worked with Rainer a little bit on some of those. I actually spent a sabbatical at Hopkins Marine Station with Gilly, who does a lot with squid and octopus. Some of the well-conserved, what's interesting is chromatophore function. This is the ability to change colors in an octopus, are controlled by different peptides and, formurfamide and others. And so you can actually apply them on the skin of an octopus and watch the chromatophores change. And yet they're under actual neuronal control to release those chemicals. And so, yes, everything in terms of the chemistry applies. In terms of the wiring, does it have to be central or not? That, decentralized processing certainly makes understanding the interplay differently. We've looked at a sea urchin a little bit, and they actually have three non-connected nervous systems, one for the feeding and the arm movement and things, and there's very little connection between them. Physiologists like to say there's none, but you actually do see chemical hormone control between them. And so there's a case where it's not, instead of calling it decentralized, there just seem to be non-connected nervous systems. And so and I think that's true of most of the brains in that particular phyla. So sea urchins and starfish are like that. And so surprisingly, the structure and the connections vary a lot, but the chemistry that controls excitability is much more stable across the different systems. So I would say, yes, everything applies. Now, I don't know, if you'll find every transmitter in every system. What you will see in an octopus, just like you'll see in a brain region, is you'll see that some are common and some are not. Only a few brain regions use serotonin, and so it could, I don't remember seeing serotonin, for example, in their arms, but there is of an octopus, but there's high levels in their feeding network, just like there is in other mollusks. Now, yeah, I don't know if the soup is chicken noodle. It's an interesting point. For a long time, people like to say the brain was wiring, and that was one engineering point of view, and the others said it was chemistry, and the two people didn't talk. And now it's becoming obvious it's both. If you have a ruler, then everything is distance, and if you have a balance, everything is weight. And so if you're a chemist, you measure chemicals, and so that's what you think is important. And if you're measuring, if you have a microscope, you measure structure. The two are very related.
So I actually had a question for you, because you kind of touched on this topic a few points. You mentioned, do you know why the gut or the digestive system has so many neurons, or why there are so many neurons in the gut compared to the brain, or why it's specifically those two areas? Because I know you mentioned.
And so I have an answer, but I want to make sure. So people make up answers to questions like that, and then it becomes dogma, and it's not necessarily true. So in other words, you know what I'm saying is, so one of the things is if you go, you look at a lot of simpler animals, mollusks and others, the nervous system evolved and was located near the feeding networks, right? I mean, it controlled feeding. And so it wraps around the esophagus and feeding. And so that's one of the ancient, so even in a human, the enteric nervous system in our gut is non-myelinated and in many ways has central pattern generators, just like you would see in an aplasia. It's a very old, it appears more ancient than the others. Now, the reason I say that is how long have we had the enteric nervous system? Just as long as the other system. So it's not really more ancient. It's just that it has many of the characteristics of ancient nervous systems. And serotonin was a molecule, for example, used as a threshold tonic for that. And so it's used, that's actually the conserved function. So 90% of the serotonin in the human body is made in the enteric nervous system. and, in the adult. The other way to look at it, though, and this is a funny way that people tend not to, is if you think about during development, when you don't have brain wiring, you have to have a couple hundred simultaneous chemical gradients because you're making body plan, you're putting not just the brain, but everything down. And every one of those chemical gradients requires a way of making the molecule. For example, the enzymes to make serotonin or dopamine or something like that, or D-aspartate, which is used then, a molecule to sense it, a receptor, and a molecule to get rid of it. So during development, you have all of these hundreds of gradients, and all of the transmitter systems are being used. Dopamine, serotonin, GABA glutamate, actually lots of peptides are used in that time frame. And there's no brain yet. It's even before there's a brain. And so it's doing chemical gradients, and then they're turned off. And then some of those are used in the more adult form as far as neurotransmitters and neuromodulators. And the reason that's important is that you can find the transmitters all being used during the body, during development. You see this in a fruit fly and others, but even in mammals, and they all go away, and then they have this role in an adult. And that means that once it's used in development, you can't get rid of it, right? So if you use serotonin in development, if you use dopamine, if you use even a neuropeptide Y or substance P, neuropeptide Y is found in Aplysia and it's found in humans. It does something different, except it's expressed at the same time during development. So once they're sort of locked into development, they're sort of in the genome and you can't get rid of them. Because if you're making an organism and you pull that out and everything falls apart, that's the end of the organism. And so I think a lot of the neurotransmitters and neuromodulators are also developmental cues, and that's what keeps them so conserved, because you can't mess with the development. And then, when you need a new signal in the brain because you're gonna, you want a whisker movement or something in a mouse, or you want something for a human, you just reach into those 200 or so that you have and you grab one out. And so I'm guessing, but again, and other people have said the same thing, this is a guess, because how would you test that? That they're locked in because of their developmental role, even though we tend to think of, people like to say serotonin is relating to feeding or serotonin is relating to depression, but actually serotonin's first role is in development. And if you knock that out, the animal's, basically dead, doesn't develop right. And so that's the function that keeps it in every animal. But that's a guess.
Thank you. That's great. Looks like we got another question in the chat here.
It's a good, it's a sort of a, I'm trying to think of, so there's two things. Is I'm not that familiar with the polyvagal theory. What I would say is that there are, if there are key chemical signaling pathways that haven't been known, and you figure out what they do and how they work, then of course that would help develop how they control. For example, there are a large number of signals that are passing, in the enteric system, in the heart, in the other organs that are used by neurons there that may, or that help align signals between them. As an example, I mentioned D-Alanine is a neurotransmitter, or it's found in islets, it's found in the brain. It turns out, as far as we can tell, the mammalian, no mammal have an enzyme to make D-Alanine. And so it's made by the microbes in your gut. It's taken up by neurons in the autonomic nervous system, and then it's used as a transmitter there. So instead of being made, so this is a very weird case. Is it a neurotransmitter? Well, it violates the rule that it's made by the system. But that would be one where this, understanding how these work actually tells us something about the system. Is it a neurotransmitter? Well, it hits the NMDA receptor. It helps modulate its activity. I don't know if it's classed, a neurotransmitter or a hormone, or how it works or how it's transported. We're studying that. And we have some grants to study that, but that's an example of where you're going from gut microbiome to the simpler systems in the body all the way up to the CNS. And we're still trying to understand the links there. So yes, I hope it will have an understanding, but I'm not sure where it'll go. You know, right now, in the cases of that, and it's something like that, we're really just at the beginning trying to understand how it even goes from the gut microbiome into the endocrine systems and nervous systems. Those studies were actually supposed to happen this year and also got interrupted by a little bit by COVID, but we're using animals that don't have a gut microbiome and then introducing D-Alanine to, you know, isotopically labeled D-Alanine to see how it flows through the system, and we'll see where that goes. Good question. But I'll have to look up a little bit more. But as I say, the link between the disparate sets of neurons in the enteric system and other systems and how they talk to each other through these chemicals is really still sort of being explored, I guess is what I would say. And so our work will actually, I guess the help would come is if we understand how that works. As far as in terms of understanding behavior, it's going to be a little bit harder to follow. Right now we're at the point trying to understand what's there and what it does. And once you have an idea, that will tell you something else. Now, the interesting thing is when you see something like D-Alanine, D-Serine, or others, there's no question that you can manipulate the levels of these by diet, whether that's good or bad. And so it may actually have, if you understand what these systems work and do, then it may be that, just like I said in the very beginning, understanding what's happening would allow you to do the manipulations required to correct if there's an imbalance. The reason I'm hesitant to say that is in many cases, people have a feeling that if something is good, then more is better. And the case of, D-Serine supplementation, the amount that's used is micrograms, that's a normal diet, and people can buy grams because it sounds better. And taking a millionfold excess of anything is not necessarily safe. And so there may very well be a therapeutic to eventually understanding of what these do, but I would actually approach this fairly slowly and keep it reasonable levels. But now I have some homework. I'm going to have to look this up. Thank you. Thank you very much. Enjoy the rest of your day. And if you come back to the Science Cafe, I guess we'll see you next year.
Yeah.
Instead of saying the end of the semester, we'll say the last one of 2020. The last one of the, or I could say, depending on how you count it, the last one of the decade.
Also true, yeah. All right.
I shut this down for a whole decade. Thank you all.
Yeah. Thank you all. Thank you for joining us, Dr. Sweedler. We'll be starting Science Cafe back up in February for anyone who wants to join. And if anyone's interested in signing up for their listserv, let me know. You can send me a message or you can put your email in the chat and I can get you on the listserv to get emails about future Science Cafes. Otherwise, again, thank you very much. And I hope you have a great rest of the year.
Bye.
Bye.
Thanks, Danny.
Yeah, thank you.
You too. Take care.